Hello friends, welcome to our YouTube channel Pharmacy World. We were in the B Pharmacy 4th semester of Medicinal Chemistry 1. We have completed 5 to 6 lectures in the last videos. So those who didn't see our last videos, I will put the link in the description box or else you can check the playlist. Today let us complete two topics which include protein binding and chelation. Starting with the first topic, protein binding. It is very important topic. Let us understand with a small story. Think this is you. Please don't mind about the diagram. I know you guys are beautiful or handsome. So, as drug is moved from the mouth, then they reach to esophagus and finally to the stomach. The stomach contains HCl, which helps in the breakdown of the drug. After breakdown in the stomach, they move to the intestine. As you all know that major absorption takes place in small intestine. Drug moves to blood capillaries from the small intestine. This is called as absorption process. Later, when the drug enters the blood, they combine with the plasma proteins present in the blood. Means the drug bind together with the plasma proteins to form a complex. So what is a complex? When two or more compounds bind together, it form a complex and the process is called as complexation. So the drug comes and bind the plasma protein of a blood to form a complex and that process is called as protein binding. Protein binding is also called as protein drug binding because it is a binding between a drug and protein. So I hope you guys understood what is protein binding. In order to make you more clear about what is protein binding, observe this. So this is a drug, this is a protein present in the blood. When drug enters to the blood, they combine with the protein and form a complex. Drug which enters the blood do not combine completely with the protein present in the blood. Some drug remain in free form. The drug which binds the protein is called as a complex form. The drug which remains in free form is called as free drug molecules or it is in free drug form. There are some types of proteins in our blood. Let me explain them. See here. So the plasma proteins present in the blood contains albumin, alpha acid, glycoprotein, lipoprotein and globulins. In blood, there are many other components other than plasma proteins like blood cells, blood protein of extravascular tissues. Mostly drug binds with the plasma proteins. In plasma proteins, drug bind mostly with the albumin because they are present in large amount. When drug is combined with the protein, they form two types of a complex. One is reversible and the other one is irreversible complex. For example, this is a drug. So this is a drug and this is a protein. I told you earlier that when drug enters the blood, they combine with the protein and form a complex. So this is how a complex formed. Here there are two situations. Either they break down and dissociate to form drug and protein separately after complex is formed, or else they remain in the same situation. If they convert again to form separately, then it is called as a reversible process. So this is a reversible. Or else if they remain in the same complex formation, they are called as irreversible. I hope you guys understood what is reversible and irreversible type of protein binding. Now, when drug and protein forms a complex, there is a force combining them, which means they involve a strong bond. Here, when these complex are formed, they have strong bond. So if there is no complex formed between drug and protein, then it have a weak bond. Strong bond involves covalent or ionic bond, whereas weak bond involves van der Waals bond and hydrogen bond. Let us see how Protein binding is related to biological action. See here, drug which enters the blood contains protein. They form a bond in blood and form a complex. So here there are 5 complex formed and other drugs are in a free form. Now the binder drugs move to the site of action. For example, you have headache. Fine, you have headache. Now the drugs move through the blood and reaches to the site of action. If there is a pain in the shoulder, the binder drugs move to that place also. So this is a simple way how drugs reaches to the site of action after protein binding. After reaching the site of action, what happens? Site of action contains cell, membrane, extracellular fluid, intracellular fluid. After reaching the site of action, the drug must distribute itself and the process is called as distribution. In order to enter the extracellular fluid of cell, the drug must pass the membrane. We had 5 complex which pass the membrane and reach the site of action. So this is the cell where the drug has to show its reaction. Let us zoom this. The site of action is also called as receptor, but the complex drug should enter the site of action in free form. First, in order to cross the cell membrane, they have to be in complex form, and later when it reaches the site of action, they should be in free form. So that protein complex formed should detach separately to form free drug in order to enter the site of action. First, drug and protein should bind in the blood. They form a complex in order to pass the membrane of the cell. Later, when they reach a site of action, they should detach from the complex and move freely into the site of action. This is protein binding and its mechanism of how it works in the body. So remember, when drug attaches with the protein and get detached from the protein to reach the site of action, it involves reversible and irreversible process. Protein binding also influences the bioavailability and distribution of active compounds because the rate of drug induced is not equal to the rate of drug reached the site of action. So bioavailability is not 100%. So if anyone have doubt, don't get confused. You can watch the video again. I have tried to make you clear what is protein binding. I hope you guys understood what is protein binding and how it is important in our body. So let us move to the next topic, that is chelation. So what is chelation? It is same like a bond formation. For example, this is a metal. It has many ligands here. Ligands. So ligands and ligands. It has many ligands here. So this ligand starts to form a bond. The bond is formed. So this way a complex is formed and the process is known as chelation. Here ligand has 2 or more donating group. For example, see here it contains NH2, NH2, CH2, CH. Here the metal ion get the lone pair of electrons from the both the sides. From here they get the electrons and from here also they get the electrons. So the metal accepted 2 types of electron from the same compound. So this type of process is known as chelation and compounds are called as chelates. Read the definition. So chelation is a type of complexation. It is a type of bonding of ions or molecules to metal ion. The compounds that are obtaining by donating electrons to metal ion with the formation of ring structure are called as chelates, and the process is known as chelation. The compound capable of forming a ring structure with a metal ion is termed as ligands. The chelating property is restricted to atoms like nitrogen, sulfur and oxygen, which are electron donating group. Let us see how chelation is related to biological action. When there is heavy metal poisoning, heavy metals includes lead, mercury, iron and arsenic. So during heavy metal poisoning, EDTA is given, or otherwise ethylene diamine is also given. So they form a bond with the arsenic. Complex is removed from the body along with the arsenic. Removal of heavy metal poisoning is called as a chelation therapy. It is used in poisoning also. Same in the case of EDTA. If there is heavy metal poisoning, this EDTA combined with the heavy metal and they are removed from the body along with the heavy metal. So I hope you guys understood today's topic related to protein binding and chelation. If you guys like the video, hit like, share and subscribe to the channel. So if anyone has doubts, please comment below. So until next video, take care. Bye bye.